Welcome. You're now going to listen to information about cell membranes, which is going to be Chapter 3, Section 3 of your textbook. This will actually cover Section 3, Section 3, Section 4, and Section 5, all of which feature kind of processes that are really involved with cell membranes. And honestly, the cell membrane is so dynamic and so important. Let's go ahead and make sure we know a lot of information about it to really explain how cells work, how they function. We've already mentioned a lot of information about phospholipids before. Just a quick review. We said phospholipids, they're a lipid, one of those macromolecules that are so important to building blocks of life. And we said they're made up of three things. There's a charged phosphate group, which you can see up here. You can also find that glycerol, which is just the kind of the bottom portion of the head. And then we can find there's two fatty acid chains. One is going to be saturated, which means there's all kinds of hydrogen stuck to it. And then we have the unsaturated fatty acid, which caused that little break out, that little odd shape to the tail, which makes it very unsaturated. It makes it actually really nonpolar as well, because we know that lipids are nonpolar. So this side actually doesn't like water, whereas the head actually does. Very cool feature. All of these phospholipids really take up a huge portion of your cell membranes. Now, we can also call your cell membrane the plasma membrane if you wish to. Some people prefer to call it that way. And basically, it acts like a little gate, like the barrier between the inside and the outside of the cell. Now, we don't just want anything being able to move in and out of the cells, and that's why we have two layers of phospholipids. The reason why is actually going to be found here on the next slide, but you'll see it basically gives us a similar feature on the inside and outside of the cell. And it means it acts the same way, which means it allows certain things in and out the same way, depending on either direction that you're moving. So we mentioned how the heads of each of those phospholipids are hydrophilic or polar, which means they bond and they form those hydrogen bonds very easily with water. And the cool thing is, since we have two layers of these on our cell membranes, or as part of our cell membranes, it actually causes those interactions to be okay. It happens all the time, and that's good. And it happens on the outside and the inside, or the extracellular or intracellular spaces. But those nonpolar tails, those hydrophobic tails, they face towards each other in the middle. And since there's this giant thick layer of hydrophobic area, water just can't easily move in, and that acts as a barrier to water, meaning water just can't move in or out of your cells randomly. It actually has to pass through certain gates, little channels, called aquaporins, which are pretty cool. But the main thing, remember guys, hydrophobic means water hating or hates water, and hydrophilic means water loving or water lover. So those polar and nonpolar terms really showed up again. So try to make sure you keep those in mind. Of course, our cell membranes are made up of all of the different kinds of macromolecules, with really the exception of nucleic acids. And the truth is, the cell membrane is actually having a role of protecting the nucleic acids found inside of your nucleus of your cells. So there are lipids found there, other than, of course, the phospholipids that we already mentioned. There's cholesterol that's found kind of wedged in between the different phospholipids, which actually strengthens the cell membrane. And of course allows for bonding of certain properties, certain molecules, to help it act differently as well. There's proteins, and there's all kinds of proteins. Just a common example for the inside of the cell, we have the cytoskeleton, which we mentioned on the last collection of notes that we had there. And of course, it plays a role in transporting materials, which we'll talk about more today. And we call these transport or channel proteins, depending on what their function is. Carbohydrates also play a role. They basically bond to the outside of the cells and actually bond to lipids and proteins, and they kind of act as identification tags for like cell recognition purposes, like identifying good guys and bad guys around your cells, or even just to identify your cell as a cell of your own body, so your own body doesn't attack it. There's actually diseases that are focused around damaging these, and it actually makes them not being read correctly, which means your body fights itself. I like this diagram because it kind of shows you just a snapshot of all the different parts and pieces involved in your cell membrane. So you can see the phospholipid bilayer is very prominent here, but you can also see all those blue blobs. They're all different kinds of proteins that you can find there. And we kind of are talking about peripheral proteins. They're just found on one side of the plasma membrane. We have the integral membrane proteins, which kind of span throughout there. You can also find channel proteins, which actually have a hole in there, and that's where we actually transport things. We also find some fun, cool shapes there. We can see a little cholesterol hidden right there, and they actually play a role, once again, in strengthening the cell membrane or the plasma membrane. And then we mentioned how there can be there's carbohydrates playing a role there, and that's what these glycoproteins and glycolipids are actually doing. They're bonding to other molecules, and they're playing a role as well. Once again, they act more as like a receptor or a identifier on the outside of cells. So there's a lot of different parts and pieces as part of the cell membrane, and we use this phrase called the fluid mosaic model to actually describe what's going on there. And by the way, like it actually is a relatively big concept for so many proteins and different portions to actually be found inside of the cell membrane. For a long time, people didn't really realize that there are as many proteins as there actually are, so it's a cool thought. Anyway, this model helps us explain why cell membranes, seem to be selectively permeable or semi-permeable, basically meaning they allow some things, but not all things, to pass through it. It's very picky. And this is really important because we're talking about your cells. And of course, if anything got in there, we could be talking about bacteria or viruses that can just come inside of your cells and wreak havoc, which would be awful. So the fluid mosaic model helps us describe all the different portions and pieces and how intricate really the design of the plasma membrane is. Let's look a little closer at this picture here, and you can see we're talking about some of those protein channels, like those proteins that are used for transporting things, and we're going to get into cell transport here in a little while. We also have some of those glycoproteins, and we can even see some of those glycolipids kind of wedged there. That's the green guys that are hiding right there. Cholesterols are also wedged in there. We can also find, once again, I like this picture because it does show us a little bit more of the actual proteins that act as our cytoskeleton, shown in the cytoplasm of our cell. And I want you to remember, guys, the cytoplasm is found inside of cells. Extracellular just means outside of the cell. It also helps us identify each of those individual phospholipid molecules, which are really important because there's thousands and thousands and thousands of them just in a small section of your cell membranes. And the last thing I also want to mention is that you can actually see really a lot of the structure of proteins that we mentioned earlier, some of the secondary structure, and you can see an alpha helix protein here playing a role in the cell membrane. So it's not always smooth, it's not always just a blob. All proteins have very complex structures, which we mentioned actually in our last unit, which is a cool thought. Also, when we're talking about those proteins, they serve that purpose of basically acting as a receptor. Now, receptors are proteins that detect signal molecules that are floating around either inside or mostly outside of your cells. We actually call these little signals found on the outside of our bodies just floating around, these chemicals, they're called ligands. And our bodies communicate to each other, or rather our cells in our body communicate to each other, basically sending ligands around. And having a certain kind of receptor is like having the right kind of receiver. It's kind of like think about having a walkie-talkie. You got to be on the right frequency, and we make sure, so we can get all the messages that we possibly can, we have a lot of different walkie-talkies stuck on the outside of our cells tuned in to different frequencies. So whenever we get a signal in one particular frequency, hopefully we'll be able to listen to it, understand it, and act accordingly. And once again, I just want to point out to you, just like when we talked about the lock and key model for enzymes, a ligand's got to be able to bind to a receptor, which is going to be a protein, and it's got to fit to that in a particular way. So certain ligands fit to certain proteins in ways, and that's why it's very important that these complex shapes, which you see up here in the upper right corner, that complex structure of the protein is maintained. So it's very, very, very important that we remember that complex shape plays a major role in what actually interacts with our cells. Let's go ahead and talk about section 4 of your textbook. That's going to be diffusion and osmosis. And I'm going to introduce you Oh yes, I'm going to introduce you to your very, very best friend. And that's called the Triangle of Love. I know. It's pretty much a work of art here. You can see there's a right triangle here, and there's a little heart inside. I want you to possibly consider getting this tattooed on you as well. It's probably the most important tool that's going to help you understanding the rest of our entire conversation today. I got a little copyright symbol up there. No, I have not actually copyrighted the Triangle of Love, but I just wanted to give the Triangle of Love some love by making him think he was very important by being copyrighted. Anyway, the purpose of the Triangle of Love is to help us describe how energy is used in our cells to move things in and out of them. So if you can remember this one shape, you are going to be able to remember exactly how things move in and out of the cells. So we use this triangle to describe something called the concentration gradient. Now, the concentration gradient is just basically the number of solutes in relation to the solution itself. So do you have a high number of solutes? That means you have high concentration of solutes, which means you would be high in that gradient. If you have a low number of solutes, of course, you're going to be low in that concentration. So that's going to be lower in the concentration gradient. So once again, if we're higher up on this triangle, that means we have a high number of solutes. And if we're lower on the triangle, we have a low number of solutes. So we basically move things in and out of cells based on the concentration gradient, how many solutes are in or out of the cell. So if we move up the concentration gradients, that means we're going to be participating in something called active transport. And the cool thing about this is it's going to do something different than passive transport. Passive transport is going to be moving down the concentration gradient. So you can see clearly here we have a slope, a gradient. That's what this ledge, this angle that you see on the triangle represents. If you're moving up that concentration gradient, let's pretend you were going up a hill, it would be very important you recognize that it would take you more energy to do that. So if you move up the concentration gradient, you are going to be involved in active transport, which requires energy, usually, by the way, in the form of ATP, which we will talk about a little bit later in the year. But if you're moving down the concentration gradient, which is passive transport, you are not going to need to use energy. It's pretty much free to use. Now this means you'll be moving from areas of high concentration to low concentration. Active transport, you're moving from areas of low concentration to high concentration. So let's try to keep this tool in mind throughout the rest of our discussion here so we can actually understand what's going on inside of our cells. So passive transport, just to define it for you, is the process of moving things in and out of a cell without using energy. Remember, guys, we do not use energy using passive transport. Now, we always have things moving in and out of our cells, but if we always required energy, we'd be tired all the time. Things have to flow through us at massive rates just in order for them to function. So this is kind of our body's way of being able to use basic physics in order to help us accomplish something so vital to our survival. Now, there's two processes we're going to talk about. They're called osmosis and diffusion. You've probably heard of these before, and they are both versions of passive transport. Now, diffusion should be very familiar to you. It's not just used inside of your cells, it's used all the time. And basically, it's the movement of materials from a high concentration to an area of a lower concentration, which means it moves down the concentration gradients. Now, this actually means that solutes, substances, will actually disperse over time, and they'll actually disperse evenly. So if we just have a container full of some kind of material here, let's say it's sugar, and I pour that into my glass, over time my sugar will dissolve, because both things are polar, by the way. They will dissolve and it will be evenly spread inside of the glass. Think like when you add sugar to tea, right? You put it in there, it might settle on the bottom as the granules, then over time with either stirring or heat, you will see it dissolve, and every sip you take was going to be sweet because it's dispersed, it's actually diffused throughout the entire glass. You'd actually probably notice this not in just like a liquid, but you can also notice this in the air. If somebody walks into a room and they're covered in perfume or cologne or a really strong body spray of some kind. I used to teach middle school. Kids used to do that all the time. They'd walk in through the door, and if you were standing next to the kid, of course you would smell them, and it'd give you a headache. but the people in the far corner of the room, they wouldn't smell the kid until like 3 minutes later, when all of the particles have, or a lot of the particles have now diffused into the air and transported all the way to the other side of the room. By the end of the class period, you smell like, you know, gross, sweet, nasty, sweaty middle schooler, which is just, what a terrible experience. I'm glad I teach high school. Sorry, middle schoolers, if I ever taught you, but you did smell bad. So over time, keep in mind, we are moving things through that cell membrane, and they do not require anything special. They just kind of passively just pass through the phospholipid bilayer, that cell membrane. And you can see over that time, we will see an even dispersion. So on the outside and the inside of cells, we'll see about the same amount. And it actually will maintain that. Let's pretend the inside of the cell also gained some magically, I don't know how, but let's say they gained a few more things that were also easily passively transported. Well, eventually those would move through and move in the opposite direction, so we could eventually diffuse that into a nice even way. So just remember, guys, diffusion goes down the concentration gradient. And I'm sure, hopefully, you have seen this before, but if you just add dye, like food coloring, to water, you'll see it slowly spreads out. You can see how dark and heavy the concentration is when the drop of dye enters the water, and it slowly spreads out. You don't even have to mix it, but it will eventually diffuse throughout the entire solution. So try to keep in mind, solutes will diffuse. That's a very natural, very common thing to occur. Now, osmosis, this is actually a form of diffusion, but we don't think about solutes anymore. What we're thinking about are just water molecules, nothing else. So we're basically moving from a high concentration to a low concentration, but we're moving from a high concentration of water to an area of a lower concentration of water. And remember, when we talked about solutions, we said water usually acts as the solvent. So if you have a lot of a solute, then you're going to have less of a solvent in that area. You know, it's just going to be they're occupying that same space. Now, this basically means if you have less of a solute, you would have a higher concentration of a solvent, and kind of vice versa. So let's go ahead and look at an example here. And this is actually what we call an osmosis tube. Right here in the middle of our tube, little U-shaped tube, this is going to be semi-permeable. Okay, semi-permeable. And that semi-permeable or selectively permeable membrane will allow water to pass through it just fine. Let's pretend there's like little tiny micron-sized holes that allow water to pass through. But if anything else larger than that is there, it will not pass through it. It'll be filtered away. So if we just fill both sides of the tube with the same amount of water, which can evenly flow throughout, and we add sugar, C6H12O6, to one side of that tube, the sugar will not be able to pass through. But we will actually create a lower concentration. Let me write that for you. Lower concentration of H2O on this side once the sugar enters that tube. And that's because we've increased the concentration of a solute. Therefore, we must have less solvent. What will happen is osmosis will take place, and water will want to move into the opposite side with the sugar because it wants to distribute it as evenly as possible, and we want to have about the same amount of concentration on both sides. So it'll kind of make up for that extra space that's now been taken up for the sugar, and the water will try to move in. So you actually see the level of water inside of the tube on opposite sides actually change. This is actually really important in like filtering water because they use this process of osmosis and something called reverse osmosis to actually filter water. So we can actually use a pretty passive process to filter things out. Now, osmosis is wildly important because, of course, water is all over the place. It's very necessary for your cells. So we have these three terms used to describe the effects of osmosis. You can have something become hypertonic, which means you have a high concentration of a solute outside of the cell. You could have something become isotonic, which means the cell is basically having the same concentration of water in and out of the cell. So there's this equal, even exchange all the time. And then there's hypotonic. And hypotonic is when we have a higher concentration of solute inside of the cell. Now, basically, hypertonic means water is going to move out of the cell. And hypotonic means water moves inside of the cell. Now, these can actually cause different things to occur to your cells. So if we look at examples of, let's say, red blood cells, we can actually see this take place. And actually, just to add to that idea, it's not just red blood cells, but also plant cells. They're wildly dependent on the osmotic pressure changes that we find in due to water. But anyway, hypertonic, once again, we said that's when you have a higher concentration of solute outside of the cell, which means that water wants to leave the cell, and this would actually cause something called crenation, which actually causes cells to shrivel up, which is not a good thing. This would make them all wrinkly and not function the way they're supposed to. Hypotonic, that's the one over here. Hypotonic means that we have a higher concentration of solute inside of the cell, which means water wants to rush in. This actually causes the cell to swell up. And in some cases, if the cell swells up too much, it causes them to burst, which we call lysis. And that's, of course, very bad too. That would actually kill the cells, and they would not be able to function properly. Isotonic just means that we have an equal balance of water moving in and out of cells because we have roughly an even balance of solutes in and out of the cells. So they stay even and nice and balanced. So down here on the bottom in the black and white photos, you can see examples of red blood cells that have been exposed to different concentrations of solutes. So, of course, we see hypertonic causing that crenation over here in area number 1. In area number 2, we see isotonic still, a nice even balance for those cells. And then area 3, we can see hypotonic, meaning cells want to swell up. Now, your cells, our cells, human cells, red blood cells in particular, want to stay in an isotonic solution. That's what they want to be ideally maintained at. But plant cells, on the other hand, actually want to maintain hypotonic. And that's because plants just have their cell walls to support them, and they actually use the strength of water to support their structures. This is why you'll see a plant wilt if they don't have enough water, because those large vacuoles inside of cells, which we talked about last section, of course need to stay completely filled so they can maintain that pressure. Another form of passive transport, not just diffusion or osmosis, is called facilitated diffusion. Now, this is actually one of those troublesome areas for kids to actually study because they hear that word facilitated diffusion and they think, oh, that sounds important. That must use energy. That's active transport. No, that's not true. It's actually still another form of diffusion. It just uses usually special proteins to help those objects cross that cell membrane, but it still moves from a high concentration to a low concentration. It doesn't use energy. So just try to remember, guys, facilitated, which by the way just means helping. It's just diffusion with help, and it just didn't require any extra energy for that help to occur. So facilitated diffusion is also a process of passive transport, and it kind of looks like this. We can see we have some insoluble materials, basically meaning they will not be fat soluble. They will not dissolve through the cell membrane or pass through the cell membrane. So they have to use these proteins, these channel proteins, and receptors usually to actually stick on the inside, which will actually activate like a gate to open. And then once that gate opens in area 2 here, we have area 1, area 2, that gate opens in area 2, we just pass it through there. And once again, we're moving from a high concentration to an area of low concentration. So that kind of movement and the lack of using ATP, lack of using energy, is actually why we call that facilitated diffusion, still a form of passive transport. Finally, we are going to talk about active transport, section 5 of your textbook, and it's going to be pretty much the opposite of passive transport. Now, active transport, just to remind you, is when you're moving up or against the concentration gradient, and we do have to use energy, and that energy source is going to be something called ATP, and ATP stands for something called adenosine triphosphate. We'll talk more about that when we get into cellular energy, basically respiration. We'll actually spend a good amount of time describing that, but basically it's going to become like the main currency of energy inside of your body, so we use it all over the time. And these processes happen all the time, all over the place in your body. So we do have many cases like sodium ion pumps or sodium-potassium pumps, where we have to move things against the concentration gradient in order to even function properly. And this is wildly important for every cell, so it's not like it's uncommon. It's just reality of life. Now, one of the versions of active transport is called endocytosis. Now, that keyword endo, meaning within or inner or absorbing, I'm guessing you can probably tell this means that we're taking in materials, molecules floating outside of the cell, inside of the cell, and that means that we're just moving things into. Now, whenever the cell membrane actually allows things to pass through here, it folds in and surrounds the objects, and actually causes that object to be surrounded by more of those phospholipids, and that's really kind of the unique thing. The amazing thing is about that cell membrane is that it's ubiquitous across the entire cell. We use the same membrane for all of our organelles. We actually called all of us with what we said, like human beings or mammals or animals. We all have what we call the word's slipping my mind. Wow. Eukaryotic cells. There we go. My goodness. Eukaryotic cells have membrane-bound organelles, which actually allows objects to move in, to cells, and we can actually unite them with the other organelles that are found inside of our body. So it's very important. Anyway, the vesicle can sometimes connect to a lysosome and help break down that material. And remember, lysosomes are usually filled with enzymes. Sounds familiar. To break things down. So endocytosis, we move things into the cell against the concentration gradient or up the concentration gradient. Two other versions of endocytosis is going to be phagocytosis and pinocytosis. Now, phagocytosis is basically when we eat large molecules, and pinocytosis is when it seems to drink small molecules. So basically, there's a bigger version and a smaller version, but it's basically the same process. Basically, we use this all the time in the immune system or just transmitting signals between nerves. And basically, we do have that hugging motion that seems to surround all the different material that we're bringing inside of cells. And this is amazing. It's usually very directed and very well guided. And I actually want to show you a picture right here. And this picture actually shows you what that kind of looks like. And I would recommend in class, I'll probably show you videos of this. It's easier to see or understand this when you actually see the movement. And that actually kind of makes sense why this form of transport takes energy, is because the cell membrane is actually modifying and manipulating itself in a pretty dramatic way to move things inside. So phagocytosis, you can see there's a solid particle that's moving in here. And remember, we have that phospholipid bilayer. It's going to fold in a particular way and kind of stretch out like two arms, and it's going to kind of draw this large object inside, and called the pseudopodium, and it wraps around it. And finally, once the little arms seem to wrap all the way around, they seem to unite again, and that forms a little bubble, which allows that object to now move freely inside. And we call this now our vacuole or a vesicle, and that can be moved around. In pinocytosis, which we mentioned, that is when we have all this fluid out there, and the same process basically occurs. Instead of grabbing outward, though, it more so folds inward, creates a little low pressure area, and those objects now get surrounded and brought inside and just dropped off inside of the body, and we're allowed to use them however we wish. I just also want to mention, you don't have to remember this one, but it's a nice thing to know. There's also a process called receptor-mediated endocytosis, and that's when we use receptors, identifiers, to actually grab particular ligands a lot of times and actually form a bond there, which actually sends a signal for this thing to actually fold inward, and then eventually we bring those inside of another kind of vesicle and we trap those materials inside. So a very smart way that the body can actually do this. Of course, this seems to be way more complicated, way more involved, which I think makes more sense that it does use energy. Instead of endocytosis, which means inside, we also have a process called exocytosis, which is quite easily described by just being exiting or outside or out of the cell. Now, it looks pretty much just like endocytosis, but it just works in the reverse order. So endocytosis is kind of the way that your cells eat things, Exocytosis is kind of the way that they poop things out, or vomit things out, if you want to consider it that way. Sorry for the gross analogy there. But basically, it's going to allow particles or molecules to be brought outside of the cell, and you can kind of see that little process right there. I'm just going to end with this picture from the National Science Foundation. It does a really good job on showing you kind of that inward folding process. And I know we've been looking at a lot of two-dimensional objects, but this picture shows it more of a three-dimensional way. If you actually look at a cell and look at the exterior of the cell, you just see the little tiny you can actually see it looks like scales, but those are the tops of those different phospholipids, and you can see what it looks like when that actually starts to fold inward. It seems to take more of a like a little cavity shape, and then up here in this picture we can actually see that familiar shape that we've already been looking at. And once that surrounds and these areas seem to bridge together and reconnect, that actually allows them to pop off like a little bubble on the inside and form those vesicles or vacuoles, and they actually transport into the cells. And then you can see the little red things on the outside can be like lysosomes or peroxisomes or whatever they are. But basically they're filled with enzymes, so hopefully they can break down the molecules that have been brought inside of the cells. So just try to keep in mind, guys, cells are three-dimensional. And they're happening, these processes are happening on all sides of them, and millions and millions and millions of times every second. We find these everywhere. So a pretty amazing thing. That is going to be the end for Unit 3. I know that was a lot of terminology, a lot of strange words. Once again, use the videos, use the notes, go through there and try to memorize those terms. Try to look at prefixes and suffixes to try to help you understand some of the true meanings of those words. And as always in class, if you have any questions or anything you'd like to bring up, just let me know and I'll be more than happy to help. Have a great one.